Okay, so I think before the long weekend, we left off here. And so thinking about this idea of protocells, ancient cells that were here, and they're saying, I'm pushing it back to around 4.2 billion years ago. And this idea that you had RNA as the primary storage, information storage molecule, shifting over to DNA over time. I just read a paper last Friday that was looking at how rainwater back then was playing an important role in the formation of protocells. And it was written by engineers uh, that, so a lot of it's was not my expertise. So I'll be honest, I didn't understand everything that they were saying in that paper, but they collaborated with several different scientists. One of them was a Nobel Prize winning researcher in evolutionary field of study. And they think rainwater was actually providing a mechanism for protocells to form and then to allow for speciation to occur. And so, again, this is new information. I had never seen that or heard about it in terms of the evolutionary process and allowing for cells to form and persist long enough that they could become separate from others and be unique, and then start to evolve from there. So lots of information, lots of people studying this process from many different disciplines. And so you're going to keep hearing more about new information coming with evolution. But this has been another big point in evolution that has met with a lot of intense study over the years. How did we go from an anoxic, no oxygen environment to one that is oxygenated? And so they often will depict the evolutionary timeline, something like this, where you have the idea here. They're showing anoxic in the kind of the gold and orange and red layers here in the timeline. And then you get the oxygenic idea coming up. And then above that line, you see the increase in oxygen percentage on the right here. And so you can see it didn't go from 0 to 20 like what we have currently. It was a slow process, primarily because, the first organisms to actually do photosynthesis and release oxygen in that process are the cyanobacteria. So they came first. It wasn't the plants that normally people teach you, oh, the plants gave us oxygen. Well, they weren't the first. They're certainly important, but they're not the first. And so, trillions of cyanobacteria all doing their version of photosynthesis, which is very similar to how plants do it. And then they release oxygen as a byproduct of that. And you have to imagine that happening. And you can see on the timeline, you have, a long stretch of time here to go from 0 to 20% oxygen in the atmosphere. And it wasn't really until we start getting to higher levels that you see the so-called modern eukaryotic cells starting to show up. And then metazoans, that's us. We fit into the metazoan idea in this terms of multicellular organisms. And then on up. So here, if you go all the way back down, you're seeing the, very, very early. And again, this origin of life idea, this is from older data. So you would have to push this down into the orangey box because they are saying, no, it's probably more like 4.2 billion years ago. And so going from anoxic to oxygenic, here again, they're kind of really separating it brown versus blue in terms of the switch over. And they often refer to this time period as the Great Oxidation Event. That's a common way that they talk about that period of time. But you see here in the brown area that you have, this one's a little bit more accurate because they're saying LUCA is, and they have a bracket here where it is, in the 4.2 range. So this one's a little bit more accurate than the last image we were looking at. And you see, yep, they're saying, yep, this is all anoxic. And then another thing that I'll point out is, again, I mentioned this last week, anoxygenic photosynthesis. So we do have other types of photosynthesis going on that don't always release oxygen. And then you start seeing the cyanobacteria forming and the idea of oxygenic. So genic means that comes from genesis, to create. So creation of oxygen during photosynthesis. So if you are living in an anoxic environment, never been exposed to oxygen, you never really had to worry about it. But as oxygen starts to creep into your atmosphere and into your surroundings, if you didn't have a way to deal with it, it was toxic. So this idea that going from anoxic to then slowly becoming more and more oxygenated, how did they do this? And again, going back to this idea where you have mitochondria showing up here on this phylogenetic tree. So again, I know I have a few other slides further on here, getting at the idea that, endosymbiosis was one way to allow for some organisms to start being able to capitalize on the presence of oxygen instead of being poisoned by it. And so when you think about this idea of the mitochondria, they were once free living ancient bacteria. We call them mitochondria now because they're part of a larger cell, but they were once free living. The endosymbiosis theory is getting at the idea that they were once ancient bacteria that belonged into the Proteobacterium phylum, doing their thing, living their free life until they got engulfed or somehow got incorporated into a larger cell through endosymbiosis. And if you were that larger cell, and if you successfully took in that ancient cell that can use oxygen, remember that's where your oxygen is being used up right now. You're cranking out ATP because you're sucking in oxygen. It's going to the mitochondria, and it allows you to make a lot of ATP. So if you're a free living microbe and all of a sudden you have oxygen around you and you can use it to make ATP, that's awesome. That's a really good advantage for you. So then if you got engulfed by a larger cell, well, that larger cell has that advantage, because if they keep you alive inside of themselves and let you do your thing, you're using oxygen to create a bunch of ATP, and they get some of that. You share it with them. And so it's a win for that larger cell. You can see how over time, instead of breaking down that ancient cell that they just engulfed and eating it, so to speak, they kept it going and kept it within their cell, and then eventually became what we now call the mitochondria. A similar situation with chloroplasts, where they were once free living ancient cells doing their own thing, living their own life until they got engulfed by a larger cell. And in this case, they're using sunlight. So if you were that larger cell and all of a sudden you have this smaller organism in you that can now access sunlight and make good use of it, again, you get a lot of ATP from taking in sunlight, doing your thing. So again, it makes sense for that larger cell to maintain the chloroplast instead of, which was once a free cell, ancient bacteria. So things like this, they are thinking about, mulling over, and starting to think about, how did we switch? How did life still persist when you go from no oxygen to oxygen? One of the things that's actually changed quite a bit over the years is going back to this phylogenetic tree. This is very abbreviated, but one of the things here is with domain archaea. When I was first learning about domain archaea, there was more to it. They've now whittled down how many phylum are in archaea. So I would like you to know, and it's only two, I would like you to know those names. It's Euryarchaeota. Okay, that's one of the phylum names. And then this other one, Crenarchaeota. Those two, there used to be, well, when I learned it, it was three. And I think before that, they had even more. So the phylogenetic trees, they keep getting refined with more and more sequencing data and comparison. better tools, technology. But right now they're saying domain archaea have 2 phylum. Okay. And when you look at these 3 phyla, or domains of life, I should say, overall, you know, the bacteria, archaea, eukarya, you have various what we call phenotypic properties, which are, you know, how do they appear? What's their outward appearance, so to speak? And so here you can see a table trying to get at that idea in terms of comparison of the 3 domains of life. And so I would like you to take some time and study this when you're studying this for the exam, because I would like you to be able to discuss some similarities and differences between them. For sure, understanding that they, bacteria are prokaryotic, of course, eukaryotic, that domain is eukaryotic. The other idea with DNA, the idea that there's circular DNA in 2 prokaryotic domains, but linear in domain eukarya. That's a pretty important concept for this class. Some of these other things we're going to get into a little bit. And the sensitivity to antibiotics here, domain bacteria are sensitive, whereas the other 2 are not. They actually use that as a way to support the hypothesis that mitochondria and chloroplasts were once free-living organisms. They actually can show that they're sensitive to antibiotics. And so that was one of the criteria that they were using. So especially the top rows down in mitochondria and chloroplasts, those are the most important for the upcoming exam. Some of this other stuff we'll talk about later. But the endosymbiosis theory proposed by Dr. Lynn Margulis, we talked about this briefly last week, this idea that the chloroplast and the mitochondria were once free-living and were engulfed by larger organisms and became incorporated and sustained within those larger cells. And when she first proposed that, it was ridiculed and scoffed at, and she had a lot of pushback. But now, for many decades, it's been accepted as the most logical way to think about this idea of going from a prokaryotic cell type that has no nucleus or other so-called organelles, and then shifting into the eukaryotic cell type. That was a huge jump in the evolutionary process. And so they often talk about it this way, where, again, aerobic, this idea of using oxygen as a terminal electron acceptor. We'll talk more about this. So chemoorganotrophs, I'll explain more about that. But basically, they're just looking at their physiology and saying, are they capable of using oxygen or not in order to make their ATP? And then again, with the idea of chloroplast. That's a phototrope type of stuff. And again, I'll get more into that, but basically that's saying that they can do photosynthesis. And so her endosymbiosis theory is really talking about the origin of the eukaryotic cell type. That's what we are thinking about. And nowadays, most freshman intro biology textbooks will have this within the pages. So she had to present, of course, evidence to support that hypothesis. And there's been more and more evidence since her PhD thesis on this. But some of the things that she was studying and using as support were that both mitochondria and chloroplasts have their own small circular pieces of DNA. None of our other organelles, other than the nucleus, of course, have that. And so that's kind of odd, got her wondering, well, why is that the case? And then both of them have ribosomes. And when you look inside and look at their ribosomes, they look like bacterial ribosomes, not what you find in eukaryotic cell ribosomes. And when you expose chloroplasts and mitochondria to antibiotics, they're sensitive to it, just like bacteria are. So again, pointing to they have an origin of they were ancient bacteria. And then cell membrane, CM, cell membrane. When you look at bacterial cell membranes and compare it to mitochondria membranes and chloroplast membranes, they're similar. And then just their size. When you look at a mitochondria and chloroplast, they're about the same size as a typical free-living bacteria. So these were some of the pieces of evidence that she was using to support that idea. And there has been, this is not a complete list, there's been other pieces of evidence. And really, when you look in the microbial world at large, you can see evolution in action. Within our lifetime, you can see it happening. And this is one example that's often given for endosymbiosis that currently happens, where you have this idea of protozoa being in, or the green algae is being engulfed by the protozoa, which is a single cell eukaryotic organism. So on the left here, you have adult, so to speak, mature protozoa, and you see all that green inside, that's an algae. So an algae is being maintained within the protozoa and being used by the protozoa for photosynthesis. Then that mature cell went through cell reproduction and had a progeny cell. And unfortunately, one of the progeny gets the green algae, the other one doesn't. And when they do this in controlled lab conditions, what happens is they show that harmful cell separation. This one, they have flagella, so they'll swim away and go find an algae and incorporate it. And so these ideas of, a larger cell taking in a smaller cell and incorporating it into them is not just happening 4.2 billion years ago or whatever, not that old, but you have it currently happening as well. And so it's this endosymbiosis theory is accepted explanation for this shift into the prokaryotic to eukaryotic cell type. So microbes were here first. Ancient microbes, they were here first. And then everything else, when they speciated and came into existence on the planet, they came into a microbial world. And I really want to emphasize that, especially in this class, of course, because we're talking about microbial ecology. But, when our species came into existence, we came into largely a microbial world. And so we came into their life and their environment. And so it makes total sense that other organisms are going to be interacting with microbes over millions and millions of years. And we still do to this day. You can't get away from it. And so, all the way down to the oldest fossils that they have record of, you can see bacteria showing up. And so this idea of evolution and natural selection, yes, you learn about those ideas in our Intro to Evolution course, and they certainly are mechanisms being used through the timeline here. And so this information, we've talked about it, I'm just putting it down kind of formally here. And so it's idea that microbes are doing this too. We have microbes that will go through cell reproduction and pass their genetic information into their progeny cells. We certainly have natural selection weighing in and influencing what's going on, and that's been going on for billions of years. And, again, if the mutation is favorable to the current environment that organism is living in, then it will be maintained. If it's not favorable, it tends to get purged, especially when you think about microbes. Microbes are really good at taking in DNA, and if it's useful, they keep it. If it's not, they'll purge it. And they can do that all their life. And so it's something that they're very good at and really does help them survive in their current habitat. Yes. Yeah, Yeah, that's a really good question because, if you take in a gene and you think about what's the composition of, well, it's DNA, it's got a lot of molecules that make it up. And so if you take that in, you're a microbe and you're like, is this helping me? And then you go, no, it's not really helping me. And then the question is, should I maintain it? That's costly, takes resources and energy to maintain it, to replicate it. And so very often microbes they're much better at it than eukaryotes at saying, it's not very useful. I don't want to maintain it. It's costly to maintain, so I'm going to purge it. And so often they will be doing that, and we'll talk a little bit here in a minute on how they do that in terms of how do they get access to this DNA. But yeah, they're very good at taking in DNA and then getting rid of it if not needed. And if they're stressed, sometimes they will purge. If you stress in a lab environment, if you stress the bacteria, you can show that they'll start purging DNA that's not necessary. So again, this is more like a visual, just thinking about microbes hanging out in an environment, slightly different from each other. They're trying to show how you can have adaptive mutation, so that green microbe picked up some DNA, let's say, or went through a mutation event that allowed it to adapt better. And then you can see they're doing it by color, where they're shifting here, and where you get speciation occurring. And so this is something that is continually happening, really, with microbes. Microbes are very good at hanging out with each other, adapting to their environment, getting access to DNA from each other, and from their environment. And so when you get into the microbial world, the process of evolution is a little bit more, the topics that you talk about, there's more topics to consider because it's not just mutation in DNA that we're thinking about. We're thinking about some other processes. And, when you take an evolution class, very often they, talk about, well, what's a species? And generally, when you think about the biological species concept that was proposed, that's the idea that you have individuals, if they're all the same species, they can reproduce with each other and give viable offspring. And so they're part of a reproductive community. So all those individuals, they could mate with each other and give viable offspring. If you have isolation of reproductive isolation going on, you can get speciation occurring. So this is just kind of the definition of biological species concept. And that's widely accepted. But when you get down into the microbial world and think about, okay, are microbes having sex? And then it gets kind of complicated because, well, not really. They don't have sex like eukaryotic organisms do. They don't produce sperm and egg with half the number of chromosomes in each, and then when they fuse, you're back to the full complement of chromosomes. You get cell division and growth until you get offspring. They don't do that. So microbial evolutionary microbiologists, when they are looking at it, they're like, okay, well, how do we define a species then? And so that has been quite a debate on how to do this. We don't have sexual reproduction with bacteria. So Lynn Margulis and her son Dorian posed this definition back in 1986 of how microbes have their version of sex. And so I would like you to memorize this, please. The formation of individual organisms that contain genes from more than a single source or parent. Formation of individual organisms that contain genes from more than a single source or parent. So what they knew when they were thinking about this definition, what they had a good understanding of already was that they knew that bacteria can share genes with each other. They just don't do it through sperm and egg fusion. They don't do it that way. And so they call it lateral gene transfer, horizontal gene transfer, because it's between two living organisms that are hanging out together. And when this was first discovered quite a while ago, they thought that it was a very unique thing and that the organisms they were studying, well, maybe they're the only ones that can do this. It's just unique to their species. And then they started looking, can other bacteria do this? And the more they looked, the more they found, yeah, other bacteria can do this. So then, initially they're like, oh, well, bacteria, only individuals that belong to the same species can do this. That was the first definition. They're like, yep, only so E. coli, you have billions of E. coli right now. So only cells that are all E. coli could do this. That's what they were proposing. And then they started studying it more and they realized, whoops, no, different species of bacteria can also do this. So for example, Pseudomonas aeruginosa is one species, Pseudomonas fluorescens is another one. They can do it. So they're different species, same genera. They're Pseudomonas bacteria, that's the genus, but they're different species and they can do this too. So they're like, oh, well, cool. Well, then some other scientists came along and said, oh yeah, guess what? Between different genera. So maybe Pseudomonas and E. coli could do this. And so you're getting this idea that you can get horizontal gene transfer from relatively unrelated organisms. They don't have to be the same species or even the same genera. And so now they're finding that you can even have this idea of horizontal gene transfer amongst individuals that belong to different domains of life. So like domain bacteria and domain eukarya, we know that they can do this, which is, one the broadest classification. When you look at the classification in domain, that's the broadest. And so they're super unrelated, and yet they're still capable of doing horizontal gene transfer. So the more they look into this, the more the data was just kind of blowing the minds of all of these microbiologists. They're like, oh my goodness, this is way more common than we thought. And so they actually use the word bacteria are so good at sharing genes with each other. So we're talking about two living microbes being able to share DNA. It's not the process where you have microbe going through cell reproduction. That's vertical gene transfer. They do that too, of course, because when they decide, okay, I want to reproduce, so to speak, that's the idea of creating progeny cells, and they'll faithfully replicate their DNA and pass that into the progeny cells. That's vertical, but we're talking about horizontal. And so they first started calling it lateral, then the more common term these days is horizontal. And so I want to briefly introduce some concepts of how they do that. I won't go into as much detail as I do in GenMicro, but you need to have a basic understanding because they're doing this within their habitats and realizing that microbes, they don't really live in isolation. We study them often in isolation on a Petri plate, but in their natural life, they're hanging out with populations of other organisms. And so it does make sense that they've acquired this ability to get access to DNA from their neighbors. So here you have a visual where this is probably the most complicated version of getting access to DNA that they've never had before. And this is, well, actually, there's quite everything, but not just one. So here you have three different ideas. You have transformation, which is being shown here. So up top on the far left there, it says donor. Green is the chromosome, circular chromosome. So visually, what they're trying to show here is that when transformation occurs, what you have happening is there's some microbe hanging out in an environment and it dies. And so here it's breaking open and releasing the DNA. Usually when DNA gets released from a dead cell, it starts to fragment, and then a nearby microbe can actually sense that DNA and build what it needs to build to take that in. And it depends on the microbe how they do that. And like I said, I'm not going to go into detail how at the nitty gritty how they do that, but there's different mechanisms. But basically, these microbes have a way of sensing that there's free DNA out in the environment, and they'll take in fragments of it. And then they can incorporate it into their chromosome. And if they start using that stretch of DNA, and if it confers an advantage, they'll keep it. Otherwise, they'll probably purge it. So that's transformation. That's the easiest process. And they've shown that microbes can do this in all kinds of environments. Conjugation is being shown on the bottom here. And conjugation is the idea where you're looking at plasmids. So plasmids, small pieces of DNA that are separate from the chromosome. That's why it's drawn here as a pink little tiny circular piece of DNA. So what they're showing here is the plasmid is getting transferred from a donor to a recipient. And you see that there's like this tunnel that's being built between the cells. So they actually do that. The donor cell can actually physically make contact with a nearby recipient bacteria. They'll build a tunnel. That the donor usually does that. And then they'll hug up to each other, build that tunnel, and then parts of that plasmid get transferred over. So this donor keeps part of that. Remember, plasmid DNA is two strands of DNA, just like the chromosome is two strands, right? So the plasmid's like that too. So they actually only give out one strand and keep one strand so that they can maintain this plasmid. You wouldn't want to give it all because usually plasmids have genes on them that are very useful. And so they'll give one strand away, then they'll make a complementary copy so they go back to the full plasmid, and then the recipient does the same. And so now that recipient has a copy of that plasmid. Here on the right, what they're showing is sometimes you can have the plasmid incorporating into the DNA. So sometimes. But big basic idea with conjugation, what I want you to focus in on is that you have a strand of the plasmid being transferred. And here it's through an actual like tunnel system that's built from donor to recipient. The last idea here is transduction, being shown on the top right. So transduction is this idea that you have a middleman or middle person, so to speak, of a virus playing a role in getting the DNA transfer from donor to recipient. And so it's a little bit more complicated. You have the virus getting into this bacteria, they're calling it the donor. And what they're trying to show is once the virus gets into that cell, it basically starts to make copies of itself. So the viral particles are replicating. And during that replication process, sometimes you'll get the donor, the bacterial chromosome, put into the virus, kind of accidentally, so to speak. And so you have all these green little snaky looking things. That's the donor bacterial DNA floating around. And so sometimes as the virus is making copies of itself, it accidentally, so to speak, takes in some of that DNA and puts it into the virus. And then when that virus breaks out of that donor and goes to a neighbor cell and injects its DNA, this DNA be largely bacterial DNA from the first donor. And so you get transfer of DNA from donor to recipient by way of the virus particle. And what can happen is very often you have a virus that has enough bacterial DNA in it and not enough viral DNA. When it injects the DNA, this recipient cell will not die. It won't be destined to die from viral infection because there's not enough viral DNA getting in here to trigger that process. Instead, there's a lot of donor DNA first bacterial cell. And so this recipient cell, because it's getting some, enough of that bacterial DNA and not enough viral DNA, it persists, it stays alive. And hopefully for the bacteria, the recipient bacteria, some of that DNA is useful. Honky. Like I said, sci-fi movies, they got nothing. Just talk to a microbiologist and they can give you loads of ideas for sci-fi movies because there's all kinds of crazy things going on. And this isn't complete. There are other methods that we now know that have been recently discovered where there's other ways to build tunnels between cells called nanotubes. And bacteria are actively making these extensions of themselves. They look like kind of like flagella, but they're not. They're these tunnel-like structures that they'll build and put out off their cell. And they'll be searching until they find a cell that they can latch onto. Then they finish building the tunnel, and they're called nanotubes. And it allows for them to transfer information, chemicals, plasmids. And so the more they look, the more they're finding other unique ways that these bacteria are hanging out with their neighbors, swapping all kinds of stuff, including DNA, but other stuff too. Questions? So as you're studying this, I think this is a good visual explaining these three overall mechanisms, but I would like you to be able to describe or recognize, if I describe it to you, that you understand, oh, that's transformation. Or if I say, hey, tell me a little bit about transformation, you can do that. Same with conjugation and transduction. So make sure that you're and in the past, I've seen students kind of mix these up, so make sure that you are staying on point with exactly what is transformation versus conjugation transduction. Right? So here they're actually showing transformation occurring. This is a cell that was lysed and all that stringy stuff is the DNA. So it is easily fragmented. Once it gets out of a cell, that DNA will fragment. And usually they say it's about 10 kilobase pair fragments. And then those get picked up by nearby. And again, there's different processes for how they do that. But basically they can sense that there's DNA and they can figure out a way to do that. And then here is a close-up of the visual showing the conjugation where there's plasmids involved. And so plasmids, they say they have non-essential genes, but important genes on them. And I'll talk a little bit more about that. Here they're just showing one plasmid per cell, but they know that many bacteria carry many plasmids. And plasmids, by the way, they reproduce, they can replicate themselves within a cell. And so you can have multiple copies of the same plasmid floating around in a cell. Plus you can have a cell having many different plasmids. And now you can start to see how easy it is to get plasmids, many different plasmids within one bacteria. And it's not that difficult to do through these processes. And so it is a really good strategy. Like I said, the donor keeps back one strand of the plasmid. because it usually is a good thing to keep that plasmid. You want to keep it. And so you wouldn't give it all away. That's too generous. You don't want to do that. So keep a strand, give a strand, and then each copy so that you're back to a full double-stranded plasmid and you're good to go. So those plasmids can be very tricky from a human perspective in the sense that here you're looking at what types of genes are carried on plasmids and what do they do for the organism that might gain access to them. And you can see here, there's some good things in the sense that many plasmids have genes that break down toxic chemicals in our environment. So that's great. You know, they're helping us clean up our environment. So this degradation of octane, camphor, naphthalene, herbicides, acetone, butanol, I mean, all that stuff is poisonous to us. And so they're breaking that down and they're actually using it for their own sources of nutrition. So that's good. They can use plasmids to produce antibiotics. So some of the antibiotics, most of the antibiotics that you've taken to date in your life have been made by Streptomyces, that genus of bacteria. And they still, that's what we use. We grow them up and we don't like synthetically make these antibiotics, many of them. We work with the living microbe. But look at down here, antibiotic resistance. That's what has a lot of medical microbiologists and people in the healthcare industry really worried about, because these plasmids have multiple genes on there conferring resistance to multiple antibiotics, to the point where there are some microbes that are so resistant to so many antibiotics, there's no way to fight them unless you go to really drastic chemicals. And so it's very worrisome. And then it can confer virulence as well, where it can be bad for humans, plants. So plasmids can be good and can be bad. Depends on what perspective you're looking at. I see. It's like when they get a plasmid in, you mean? Yeah, I mean, they'll start expressing the genes, and I mean, it's a pretty quick process compared to like our bodies, but yeah, they can pretty quickly, through essentially turning those genes on. It's pretty much they'll try it out and see what happens, and then if it's a good thing, they'll maintain it. So like when you walk into a hospital, that environment, there's probably many bacteria that are like, ooh, I like this plasmid because it's conferring resistance to all these antibiotics that are in the hospital. So if you are in an environment with lots of antibiotics, like a hospital, these bacteria are like, ooh, yes, I like this plasmid. I'm keeping it because it makes me resistant to the antibiotics. So we'll talk more about that idea as we get into where microbes live. But okay, I guess we got to stop there. And we'll finish this off tomorrow. Then I'll start getting into a little bit of ecophysiology, I guess would be the way to describe the next topic.